All right, guys, gals, let's get started with our discussion on the second part of our presentation. So we just finished a discussion on DNA replication. Now that we've replicated the DNA, we can duplicate or replicate the cell. And we're going to focus on the cell cycle during this presentation with a heavy emphasis on the mitotic phase, in particular mitosis of the mitotic phase. So when we look at our cell cycle, this is the entire lifespan of the cells. We go from the parent cell to the daughter cell. And when we think of the entire cell cycle, this includes interphase, interphase being the green part of this wheel. Oh, wrong button. There we go. The green part of this wheel right here. And then mitotic phase being kind of the buff or salmon colored part of this wheel. And, you know, when we talk about the mitotic phase and cellular reproduction, mitosis and meiosis and all these terms, there's the common English usage of the word, and then there's the technical science or biology usage of the word. We're going to be using the technical meanings of these words. And I'll help to differentiate and tease out the technical meaning from the common English meaning. So during the cell cycle, or particularly during interphase, there are three different parts that we go through. We have G1, S, and then G2. When we look at this G1, we're going to make the cell bigger. During S phase, we duplicate the genome or duplicate the DNA. And during G2, we're going to have more growth as we get the cell, we continue to get the cell ready. And then during mitotic phase, we have multiple subphases. The four subphases most people care about are prophase, metaphase, anaphase, and telophase, listed right here. Prophase, metaphase, anaphase, telophase. Those can be grouped together as mitosis, and then after mitosis, we have cytokinesis. And so we have those four subphases of mitosis plus cytokinesis making up the mitotic phase. So let's talk about the phases of the parts of interphase, the G1, S, and G2 phase. This first gap, or G1 stage, is from the moment the cell is made to the moment the cell starts to replicate its DNA. And this is kind of like hey, this is the cell that needs to grow and learn to be independent, so to speak, and be able to be self-sustaining. It gets bigger, it accumulates more inclusions that have more storage, more macronutrients being stored. It's going to replicate organelles. It's going to get ready for DNA synthesis. And once it's ready for DNA synthesis, it enters the synthesis phase, or S phase, where it replicates the nuclear DNA. And I need to emphasize nuclear DNA. There is some DNA that's stored in the mitochondria, called mitochondrial or mtDNA. Mitochondrial DNA is replicated as the mitochondria replicate themselves. We as human beings don't have any chloroplasts, so we'll not talk about that DNA, but we do have nuclei in our cells, and those nuclei have the bulk of our genetic information, or DNA. During S phase, not only do we replicate the DNA within the nucleus, but we also replicate the centrioles. And recall, the centriole is that structure that makes the microtubules that we use to help move the chromosomes around during mitotic phase. After we replicate the DNA, we go to the second gap phase, G2. And this G2 stage is going to be proofreading the DNA. We'll edit the DNA, make sure it was replicated correctly. There'll be some more growth. We'll make some more enzymes. This is where we put the finishing touches on replicate the interphase before we enter mitotic phase, before we start to actually divide the nucleus and divide the cytoplasm of the cell. There are a couple more stages. We have M phase for mitotic phase. The cell is going to replicate its nucleus during mitotic phase. This is known as mitosis, so you might want to circle this section of your notes. Replicating the nucleus is mitosis, and then pinching the cell in two to form two new daughter cells, kind of pinching off the cytoplasm, that is cytokinesis. And this is the technical meaning of the word. Technically, in a biology class, mitosis is replicating the nucleus of the cell, and cytokinesis is pinching off the cell membrane to separate the cytoplasms. So that happens during the mitotic phase. We also have an alternative to the G1, G2, S phase, and that's G0 or G0. And when we look at this gap phase, this is when the cell is going to be put into stasis, so to speak. It's not going to die, but it's not going to continue reproduction. Cells that enter this G0 or G0 phase are usually very long-living cells. Think of skeletal muscle fibers or skeletal muscle tissues, cells, or neurons, or other cells of the body that have a life cycle that lasts for years. Now, as we look at this cell cycle, some cells have really rapid cell cycles, particularly cells of the tissues of our body that are damaged frequently. So if we think of the cells of the skin, the dermis, and the epidermis, these are cells that are going to be replicating rapidly because we're rapidly destroying those cells as we have abrasion and damage occurring to our body. Other cells have a slower life cycle. Think of such as adipose tissues, or perhaps some of the neuroglia of the nervous system. These are cells that can reproduce, but since we don't damage them frequently, we don't reproduce them quickly. And then there are other cells that almost never reproduce. These are cells with a life cycle of years. Think of the skeletal muscle fibers or the neurons of the nervous system. So let's talk about the mitotic phase. During the mitotic phase, there were those two key steps. We're going to have mitosis and cytokinesis. And we'll talk about mitosis first. So during mitosis, we are going to divide the nucleus. I need to emphasize this again. The primary goal of mitosis is to duplicate or replicate the nucleus and all that genetic information within the nucleus. So when we think of mitosis and cellular reproduction, depending on the textbook you look at, it's really easy to get hung up. Maybe students will spend time thinking about, is it diploid, is it haploid, is it tetraploid, and you can get bogged down in the details. And since this is not a genetics class, we're not going to spend an inordinate amount of time counting the ploides and counting the tetrads and looking at all the minutiae. We're being very pragmatic right now with our approach to mitosis. The primary thing I want to emphasize to you is that one parent cell produces two genetically identical daughter cells. The one parent cell has two copies of the genome. It's diploid. And the two daughter cells will also have two copies of the genome. They are also going to be diploid. What do we use mitosis for? And the cellular reproduction, we're going to regenerate tissues and organs, we're going to grow in volume and size as we regenerate, as we add to our tissues and organs. As we have damage occur to our tissues and organs, we can repair that damage, either the individual cells that occur, or maybe there's some gross damage. Maybe you fall off a bicycle and skin your knee, or you have surgery and there's some incision sites where there's a lot of damage. During mitosis, and again, I'll emphasize this again, it's a key point. Mitosis is replication of the nucleus. The four stages of mitosis are prophase, metaphase, anaphase, and telophase. And we're going to talk about each of those phases in detail. So first, let's talk about prophase. During prophase, we need to take that chromatin, that loosely coiled DNA that hasn't condensed yet, and we're going to cause it to tightly wrap around itself to form a chromosome. So we go from chromatin to chromosomes. The DNA goes from loosely coiled to tightly coiled. Something else that's going to happen during prophase is that we'll be able to see individual chromosomes within the nucleus of the cell. Why is it important that we tightly pack the material? We need to tightly pack that genetic material so it's easier to handle. It's kind of like, traveling at the airport. If you had all of your clothes and loosely hanging in your arms, you'd probably lose a couple pieces of clothing during your travel process. But if you take all of your clothing and pack it into a suitcase, it's a lot easier to keep track of everything and not misplace anything. The same is true of the genetic information in the cell. We take that genetic information, pack it tightly into chromosomes, and then it's easier for our cell to handle. A key step that happens during prophase is the envelope of the nucleus disintegrates. So all of a sudden, the boundary of the nucleus is gone. And those chromosomes within the nucleus are now exposed to the cytoplasm directly. The centrioles that have reproduced during S phase are going to sprout microtubules, long parts of the cytoskeleton, the spindle fibers, or microtubules, from the centrioles, push the centrioles apart, and some of those spindle fibers or microtubules are going to attach to the chromosomes at a very specific spot of the chromosome. The middle of the chromosome, called the centromere, where there's a special protein called a kinetochore. So let's skip ahead here. Here is prophase. So here we have the centrosomes or centrioles. They're sprouting microtubules. Those microtubules start to grow. The nuclear envelope begins to disintegrate. And as we look at the chromosomes, the chromatin condenses into chromosomes. In an actual photomicrograph, I believe this is of a whitefish blastula, you can see that darkly stained region. That's the chromosomes that are being tightly coiled. And you can see right here is this kind of like this star, or this point in space where there's a bunch of lines radiating out of it. That's one centriole, and then over here is another centriole. After prophase, we have metaphase. And during metaphase, all the chromosomes are going to be lined up along the center of the cell, along the equator or midline. I think of middle for meta, M and M. When the chromosomes are in the middle, you are at metaphase. The word analogy I use for prophase is pro means beginning. Prophase is the beginning phase of mitosis. So those chromosomes are going to have their mitotic spindles fully attached to them. Shorter microtubules are going to bind to the chromosomes and then will align them along the center of the cell. And this is a good stopping point once we cover this last slide. So here we have the chromosomes lined up along the midline of the cell. We have two centrioles at the opposite poles of the cell, and each chromosome is going to have a microtubule attached to it. Those microtubules are going to play a very key role in the next stage of mitosis, known as anaphase. So, guys, gals, as you're watching this video, make sure that you have your PowerPoints open and you can fill in the blanks. Like right here, here's a blank on metaphase. The notes you have should have that blanked out. And if you have any questions, please shoot me an email or come by my office hours. Happy studies.